Hello everybody, welcome to week number 5. So today we are going to talk about DNA technology. Okay, so this week we are going to talk about DNA technology. So for this lecture, there's no chapter in the book, so for this you have to follow the PowerPoint. So before we start talking about DNA technology and what is its application in different fields, um, let's go and refresh our memory about the DNA. So we have already talked about DNA. We just talked in your fourth week. We discussed the different biomolecules, and we talked about different biomolecules, and DNA was one of them. So let's refresh our memory. So basically, as you know, we have already discussed DNA is a polymer, which means it's made of monomers. And what is a monomer in a DNA? It's called a nucleotide. And each nucleotide is made of sugar, one sugar, a pentose sugar, a phosphate group, and there are 4 nitrogenous bases, and in every nucleotide there is one of these nitrogenous bases. So there is a pentose sugar, which is deoxyribose sugar in DNA, because if you remember, deoxy means there's one oxygen less in DNA, and the sugar in RNA is ribose sugar, and it has phosphate group which is negatively charged, and DNA can have these nitrogenous bases AT and GC. And in RNA there is no thymine, instead of thymine there is uracil. So this is the nucleotide of DNA. And as you know, the DNA is double stranded. So the two strands of DNA, they're linked with each other. And how they're linked with each other? By complementary. Because how they link with each other? The linkage is between the nitrogenous bases. A always links with T, C always links with G, and these are the complementary base pairing rules. So the two strands of DNA, they link with each other through these on the basis of the complementary base pairing rules, and the linkage is between these nitrogenous bases. So this is just DNA. So this is just sugar. Okay, this is the sugar present in DNA, and you can see it is deoxyribose. So at this position there is only hydrogen, there is no oxygen here. So there is one oxygen like extra here in ribose sugar, which is present in RNA. So in ribose sugar, which is present in RNA, here there is a OH group. So here is one oxygen and H is there, so it makes a OH group. But in DNA, because it's a deoxyribose, there's only H. And this is the pentose sugar. So this is one carbon, second carbon, third, fourth, and to the fourth one is attached the fifth carbon, and to the fifth carbon is attached this phosphate group, and this phosphate group is negatively charged. And this at position number 1 are attached the nitrogenous bases, and this nitrogenous base at position number 1 can be either of these 4, and these are adenine, thymine, guanine, and the cytosine. So out of these 4 it can be any. Okay, and DNA, as you can see, it's double stranded. You can see here this is the one strand, this is the second strand. A from here is linked with T of the second this strand. G of this is linked with the C of this strand. If T is here, then A is here. Okay, so this is the basic structure of DNA. Now how does DNA makes its copy? So we need to, before we can understand DNA technology, because I'm going to discuss with you gene cloning, how genes are cloned, how animals are cloned, and how can this technology be used for forensics. So before that, we need to know briefly about how does DNA make its copy, which is how does DNA replicates itself, or how does DNA duplicates itself, and how does DNA makes its protein. So we have discussed about protein expression. So briefly I will again go over. So how does DNA makes its copy? So basically this is the mechanism how DNA makes its copy. So this is one molecule of DNA and it is double stranded. Okay, so the very first thing which takes place is, with the help of the enzyme called helicase, these two strands of DNA they separate. Okay, so these are the two strands of DNA, they get separate, So this strand goes this side, this strand goes this side. So each strand now acts as a parent strand. Now with the help of another enzyme called DNA polymerase, uh, a new strand can be made. Okay, with the help of another enzyme called DNA polymerase, a new strand or a daughter strand can be made. And how does that daughter strand is made? It is made according to whatever the information is here in the old parent strand. And similarly, this is the second, this strand is now acting as a parent strand. So again on this strand, with the help of the DNA polymerase enzyme, a new daughter strand can be synthesized here. And how it's going to be synthesized? It will be all on the basis of the information which is carried in this strand. So what is the information? Information is basically the sequence of DNA. And what is the sequence of DNA? It is the order of these nitrogenous bases. So which means that if this is the parent strand and this is the parent strand, let's see how the daughter strands are formed. So in the parent strand, if the nitrogenous base is G, the new nucleotide with the nitrogenous base C will be recruited here. If it is T here, it will be A. If it is A here, T, G, C, C, G. If it is T here, A. So this will make a new daughter strand. And this is the another parent strand. So the daughter strand, which will be formed with the help of enzyme DNA polymerase, that strand again will be formed on the basis of the information which is in this strand. If it is C here, in the new daughter strand it will be G. If it is A, it will be T. If it is T, A, C, G, G, C, A and T. Okay. So you can see here, this is the new DNA strand, and this is the another DNA strand which is also new. So in the process of DNA replication, you start with one molecule of double-stranded DNA and you end up with two molecules of DNA. Okay, you end up with two molecules of DNA and both of them are double-stranded. Both of them are double-stranded. And you can see here, you can see this is the parent strand from where we started. You can see the sequence here, G, C, T, A, T. Now you can see this strand and this strand. You can see that both these strands, they are identical to each other. You can see GC, GC, TA, AT. So basically the information in DNA is all in its sequence. And what is the sequence of DNA? You must have heard this term DNA sequence. What is DNA sequence basically means? Like you must have heard like human genome has been sequenced, or the scientists, they have sequenced this thing. What is the meaning of sequencing? Sequencing basically means what is the order of these nitrogenous bases. So our genes are made of DNA, and how we are different from each other? It's all on the basis of the order of these nitrogenous bases. So if there is a difference in the order of these nitrogenous bases, it makes one gene different from the other gene. So you can see here, this is the sequence of this DNA molecule and this is the sequence of this DNA molecule, and you can check with this the parent strand from where we started. You can see this strand and this strand, they are identical to each other. They're genetically identical to each other, means the sequence is same, and also their sequence is same as the parent strand. So this is the process how DNA replicates. First of all, these two strands separate. Okay, the separation is with the help of enzyme helicase. So these two strands separate, both strands act as the parent strands, and the new daughter strands are going to be formed, and those two daughter strands which are going to be formed, they will be formed with the help of another enzyme called DNA polymerase. And how those strands are formed, what sequence they will have, it will depend on the sequence in the parent strand. And the result of DNA replication is, from one molecule of DNA which is double stranded, two molecules of DNA will be formed which will be double stranded, both of them, and they will be identical to each other as well as to the parent strand. So this is the DNA replication, and the DNA replication is called, it's semi-conservative. It has a semi-conservative mode of replication. Semi-conservative mode of replication, why? Because in each molecule of DNA you can see one strand is old and the The second strand is new. The second strand is a daughter strand. So in both of them, like in this strand, this is the parent strand. In this is the parent strand. This is the new, and this is the new. So it will happen in every cycle of replication that when the new molecule of DNA will be formed, it will have one strand which will be the old strand. One strand will be the parent strand, and the other strand will be the new strand. So that is why, or new strand or the daughter strand. So that is why DNA replication is called that it has a semi-conservative mode of replication. So this is all about how DNA replicates. So you can see this is one molecule of DNA. These two strands are separate, and so these red ones are the old strands, the parent strands, and the blue strands are the daughter strands which will be formed. And this is the way from one molecule of DNA, two molecules of DNA will be formed. So this is DNA replication. And these are the enzymes which are involved. Now, if you remember, we also talked about how DNA forms the proteins, because DNA is just the information, okay? It has the knowledge. But what does that knowledge do? It makes proteins, okay? And it's the proteins which are doing the majority of functions in our cells. So proteins are enzymes, proteins are receptors, proteins are transport molecules, proteins, hemoglobin is protein, hormones are proteins. It's the proteins which are doing majority of the functions. So our DNA basically makes proteins, okay? It codes for proteins. And how does that we have discussed that DNA makes a mRNA, which is a messenger RNA, and that messenger RNA is formed in nucleus. And you remember, nuclear envelope has the pores, and this mRNA can move through these pores, and it will go and look for the ribosomes. And what are these ribosomes? These are the protein factories. They will go and look for the ribosomes, and then they will on the ribosomes, proteins are going to be formed. And how those proteins are going to be formed on the ribosomes? With the help of the another kind of RNA which is called the transfer RNA. And this transfer RNA basically it basically transfers the amino acids. And how it's going to transfer the amino acids? That transfer of amino acids is on the basis of the information in the mRNA. And from where does this mRNA is carrying the information? It's carrying the information basically from the DNA. So this is the way how DNA makes proteins. So now let's see what is DNA technology. You must have heard about like genetic engineering or recombinant DNA technology. So let's go and see all these things now. So we have now the information about DNA structure, how does DNA makes its copy, how does DNA makes the proteins, and these are the most important things to know. So the very first thing I'm going to talk in DNA technology is gene cloning. So the very first thing, what is the meaning of a clone? Clone is something, just a copy of something, right? Clone means to make a copy of something. So you can make a copy of a gene, you can make a copy of an animal, or you can make a copy of a human. So cloning basically means to make to make an identical copy of something. So in the very first part, we are going to talk about how genes are cloned. Okay, how genes are cloned. The very first question is why genes should be cloned. Okay, so just a gene, why a gene should be cloned? So basically your genes are made of DNA, right? And this DNA codes for proteins. Now there can be some genes which are coding for some proteins which can be very useful to us, like they can act as some medicines, they can act as some vaccines. So that is why genes are cloned, because if we clone the genes, that has a lot of application, because those genes are going to code for proteins, and those proteins can be used as vaccines, they can be used as medicines. So that is why gene cloning is done. So example here is a human growth hormone. Okay, so human growth hormone can be made by gene cloning. Insulin can be made by gene cloning. Various kinds of proteins can be made, and they can be used as medicines or in vaccines. Okay, so in the gene cloning, basically we can move genes from here and there. Basically, in the gene cloning, when we have to make the identical copy of a gene, we can move genes from here and there, which means you can take a human gene and you can put in a bacteria. Okay, so you can take a human gene and you can put in bacteria. Now there's a reason why we are putting in bacteria. The reason is bacteria grows very fast. Okay, so when the bacteria grows fast, because now the gene is part of the bacteria, the gene is also going to be multiplied faster, which means that gene will be multiplied faster, and that gene will be making a protein. So because the bacteria grows that fast, if we start with 1 or 2 cells of bacteria, in the morning we will have millions of bacterial cells, and every bacterial cell will have that gene, and that will produce the protein. So we will have large amount of protein. But when we move genes from one species to another species, those kind of organisms are called transgenic organisms. Example is like if you have a human insulin gene. Now what does human insulin gene do? Human insulin gene makes a protein called insulin, and insulin is used for what? It's given to the diabetes patients. So earlier insulin used to be extracted from animals. So people who were working in these pharmaceutical companies, when they were like extracting from animals, like they were extracting very little amounts, so that the because of that the cost of the medicine was very high. But now because of this DNA technology, the gene for insulin can be cloned, and that gene is going to code for insulin protein, and that protein can be collected and it can be given to patients as a medicine. So because of that the cost comes down. So when you have a human gene, if you have a human insulin gene and you are going to put it into bacteria, then that bacteria becomes a transgenic organism. A transgenic organism is an organism whose genome has stably incorporated 1 or more genes from another species. That is the meaning of a transgenic organism. So many biotechnology products are produced within transgenic organisms. So basically you can move genes from one species to another, and that is what is the meaning of a transgenic organism. Okay, before we actually start learning about this process, how genes are cloned, we need to know a few terms. Number 1, restriction enzymes. Now what are restriction enzymes? Restriction enzymes are proteins derived from bacteria that can cut DNA in specific places. So you can just imagine a restriction enzyme is an enzyme, and majority of the enzymes are protein in nature. So you can imagine restriction enzyme is like a scissor. Now scissor can cut things, right? So restriction enzyme has a property that it can cut DNA, and it cuts DNA at a specific sequence. There are thousands of restriction enzymes available, okay, and they are extracted from bacteria. So restriction enzymes basically are naturally present in these bacteria. And why they are present in bacteria? Because these bacteria in nature, they are invaded by viruses. So nature has given these enzymes to the bacteria so that they can protect themselves, because viruses, they invade the bacteria. Okay, they invade bacteria and they can infect the bacteria. But nature has given these scissors, these restriction enzymes, to bacteria so that they can protect themselves. So what happens when the virus infects the bacteria? Because bacteria have these enzymes, they can act as scissors. They can cut the DNA of the virus and they can protect themselves. So what scientists they have done, they have extracted these restriction enzymes from the bacteria, and these enzymes are available commercially. You can buy these research labs, they buy these, and with the help of these enzymes you can cut the DNA. Okay, so they are scissors, they are like scissors, and what do they cut? They cut DNA. And third thing important is they cut DNA at a specific place. It's not that they can cut DNA at any place. No, they are very specific where they cut the DNA. Okay, this is the first term we need to know. What is a restriction enzyme? So here you can see this is your DNA. So like you can take the example of enzyme which has a recognition sequence GG8TCC. You can see here it's going to cut here. Okay, it's going to cut, basically it cuts at this point. Okay, so it is cutting here GGATCC. So it's going to make a cut here, and because DNA is double stranded, it can also make cut from the other side. It can make the cut here, and it can make the cut here also, and it can make the cut here also. So these are the restriction enzymes. Now next term we need to know what are plasmids. Now plasmids are also present in bacteria. If you remember, bacteria are prokaryotic organisms, and these prokaryotic organisms, bacteria, they have only one single chromosome. Okay, and they don't have nucleus, so they have only one single chromosome. But these bacteria, they have some extra chromosomal DNA. Okay, so it's very important to know this bacteria, they have one single chromosome, but in addition to that one single chromosome, they do have extra chromosomal rings of DNA, and those rings, they are circular, they are called plasmids, and they carry very important genes. Okay, so plasmids are small extra chromosomal rings of bacterial DNA that can exist outside of bacterial cells, and that can move into these cells through the process of transformation. So basically you can extract plasmids from the bacterial cells. There's a protocol by which you can do that, and you can also put these plasmids back into the bacterial cells. Okay, and that process is called transformation. So plasmids are extra chromosomal DNA. You can take them out of the bacterial cell, and you can also put them back into the bacterial cell. Okay, so this is like your prokaryotic cell bacterium. This is a single chromosome. This is the plasmid. Okay, it's small circular extra chromosomal, which means this is a chromosome. It is not part of this, it's separate. These are plasmids. Now the goal of cloning is that you want to make the identical copies of some gene. For example, you want to clone a human insulin gene. What are you going to do? You want to make identical copies of the human insulin gene. What are you going to do? First of all, the very first thing you have to do is you have to take that gene out of the human genome, right? So that gene belongs to humans. So we need to first of all take that gene out of the human genome, and then we have to put it into the bacteria. Because what we are trying to do, we are trying to make the identical copy of this gene, because we want large quantities of the protein, right? And for getting large quantities of that protein, we are going to clone the gene, which means we are going to make the identical copies of the gene. So for that, we can put them into bacteria, because bacteria grows very fast. But now the question is, first of all, how to extract the gene through the from the human genome. You can use restriction enzymes, because they are scissors. They can cut the DNA. So you will cut the human DNA of your interest with the help of the restriction enzymes. And now next thing is, okay, first if you have taken out that gene, now how are you going to make it go into the bacteria? You can make it go into the bacteria with the help of the plasmid DNA. Now remember that plasmid DNA is circular. How your gene is going to go into this plasmid? It cannot go. If it is circular, it cannot go. How can you make it go? To make that happen, you have to again cut the plasmid with the restriction enzyme. The plasmid is, like you can imagine, plasmid is like your rubber band. Okay, it's round. If you want to attach something to that rubber band, you need to open it, right? You will take the scissor and you will cut it. The same way you can cut this plasmid, and this plasmid is going to become, it's going to straighten up, or scientifically we call it, it becomes linear. And then you can introduce the gene of your interest, and then you can seal that gene in this plasmid with the help of the enzyme called DNA ligase. I will show you everything. So basically you use the restriction enzyme, you cut your gene of interest, you take the plasmid which is circular, you you cut it. Okay, you cut the plasmid, you make it linear, you straighten up, and then you introduce your gene of interest into this plasmid, and you seal that gene in this plasmid with the help of enzyme called DNA ligase. That results in the formation of a plasmid which is called a recombinant plasmid, because this plasmid has now your gene of interest or a foreign gene, or in your case if you're cloning insulin gene, this plasmid has now an insulin gene. So this kind of plasmid is called a recombinant plasmid. So what is a recombinant DNA? Two or more segments of DNA that have been combined by humans into a sequence that does not exist in nature. So that's what you are creating. That's what a scientist is creating. So once plasmids have had human DNA spliced into them, the plasmid can then be taken up by bacterial cells through the transformation. So once your recombinant plasmid is ready, now the goal is to make that enter into a bacterial cell. So I have already explained you that you can extract these plasmids from the bacterial cells, and you can make them go back into the bacterial cells with the help of the process called transformation. So this recombinant plasmid, which has a foreign gene, which has your gene of interest, which has an insulin gene, that's a recombinant plasmid. You can make it go back into the bacterial cells with the help of the technique called transformation. Okay. So as the bacterial cells will divide, this gene will also multiply, and because this gene in every bacterial cell is going to make a protein, so you are going to have a lot of protein. So let's see, this is a way how gene cloning is done. So here is your human cell which is containing the gene of your interest. Suppose this purple thing is your gene of interest. So this is the chromosome where your gene of interest is here. Now you have to cut it out. How you're going to cut out? With the help of restriction enzymes. Okay, this purple part is the gene in which you are interested. Okay, so you can cut it out, and how this is your plasmid. Okay, you can extract them. So plasmids are present in the bacterial cells. You can take them out. Okay, you can extract them. So this is the plasmid, and then because plasmid is circular, you can cut it again with the restriction enzymes, and then it becomes linear. Okay, it has now opened up, it is cut now. Then you can introduce this gene, you can mix this gene and this linear plasmid, and you can use the enzyme called DNA ligase here, and that will, so you will take this gene and you will take this plasmid and you are going to mix them, and you are going to add the enzyme called DNA ligase, and it is going to make a recombinant plasmid, which means it is going to with the help of the DNA ligase, this gene can be sealed into this plasmid here. Okay, this is the recombinant plasmid which is made, and you can introduce this recombinant plasmid into the bacterial cells. Okay, so you can introduce this recombinant plasmid into the bacterial cells. So once the this plasmid with this gene is inside the bacterial cell, and that happens with transformation as the bacteria will grow. So like some bacteria, they have a very short generation time. They can multiply in only 20 minutes. So if you have one bacterial cell, in 20 minutes it will become 2. In another 20 minutes, from 2 to 4, 4 to 8, 8 to 16. So this is a chain reaction. It will keep on happening. Every bacterial cell will have this insulin gene, and in every cell, insulin gene is going to make insulin protein. So if you start with one cell at night, because bacteria grows so fast, in the morning you will have millions of bacterial cells, and in every cell your gene will be there, and in every cell this gene will be making a protein, and then there are ways by which you can extract the protein, and that is, so the purpose is solved, because the gene cloning is done to have lots of protein, and that protein can be used for a medicine, or it can be used for making some vaccine. Okay, so this is gene cloning. So by gene cloning, you clone the gene, you make the identical copies of the gene. So this is just one gene, okay, which you just introduced in one bacterial cell. But because this bacterial cell grows so fast, every bacterial cell will have the identical copy of this gene. So this is gene cloning, and in every bacterial cell this gene will make the protein, and there are different ways by which we can extract this protein, and that protein can be used in the to give it to patients. So this is gene cloning. So large number of medicines and vaccines are produced today through transgenic biotechnology. Transgenic food crops are planted in abundance today in the United States. Okay, the next technique we need to learn is animal cloning. So we are going to see how the sheep dolly was cloned. What is the technology by which animals are cloned? Okay, so we just learned how genes are cloned. So it's just one single gene how it's cloned. Now we are going to learn how animals are cloned. Okay, so we know what clone is. Clone is a genetically identical copy of a biological identity. It can be a cell, it can be a gene, it can be animal. Anything can be cloned. So for cloning the animals, this technique is called reproductive cloning. So reproductive cloning is the process of making adult clones of mammals of a defined genotype. Dolly the sheep was a reproductive clone. And how the sheep dolly was cloned? By the process called somatic cell nuclear transfer. I'm going to discuss this process. So in reproductive cloning, the way how animals are cloned, they're cloned by a technique called somatic cell nuclear transfer. So what is done in this technique is, like, basically, now the aim is to make a clone. The aim is to make a clone of an animal. Like sheep dolly was clone of her mother. Okay, so it's very important to know one point here. This is the way how animals are cloned. So when you are cloning, you're making identical copy of the animal, right? Now in nature, the way animals reproduce, you need to understand how this cloning is different from the natural way of reproduction. So in nature, okay, in nature, these animals are produced by sexual reproduction. So in this process, there are two parents involved. Okay, so the offspring which is going to be formed, because in sexual reproduction, now sexual reproduction is different from cloning. So in cloning, you are making something genetically identical, something identical of something. Okay, how it's different from the sexual reproduction? Because in the sexual reproduction, there are two parents involved. Okay, so their gametes are fused. The sperm fertilizes the egg. So in that process, there are some genes which are coming from the father's sperm, some genes coming from the mother's egg. So the genes are there in both the gametes, and then the offspring is formed when the sperm fertilizes the egg, and that gets converted into a zygote, and then zygote multiplies. So basically, in the sexual reproduction, the offspring which is formed has the genes from two parents. Okay, it has genes from two parents. So that is why the offspring which is formed is not a clone of either parent, because there are two parents involved and there is mixing of the genes. So that is why the new offspring, the new baby who is formed or who is born, has the characteristics from both the parents. So that is not cloning, because that is the normal process. That's a sexual reproduction. So you must have seen that you have some characteristics from your mother, you have some characteristics from your father. So it's all because the genes are from two parents get mixed. But here's the difference between cloning is to make an animal or any organism basically who is just identical to one parent. So in the reproductive this cloning, animal cloning, only one parent is involved. So what was done when the sheep was cloned? Those scientists who cloned Dolly, they took the udder cell. Okay, udder cell is a somatic cell because their aim was to make an animal who is the clone of the mother, right? They wanted to create something who is genetically identical to the mother. So they took this udder cell or the somatic cell from the mother of the dolly, and they took from the another sheep, they took the egg cell. Okay, egg cell is a gamete which is involved in the reproduction. But what they did, they took away the nucleus from here. Okay, they removed the nucleus. They removed the nucleus because the genetic material from two cells should not be mixed, because the idea is to make a clone. Clone can be only made when only the genes of one parent are involved. Okay, so that is what they did. They took the udder cell, the somatic cell, from the parent. So that cell has nucleus and nucleus has the DNA. They took the egg cell, which is a gamete, from the other sheep, and they removed the nucleus. And the idea to remove nucleus was they did not wanted the mixing up of the genes of two cells of the two parents. Okay, so from here they remove the nucleus. When they're removing nucleus, they're removing DNA. And then what did they did? This cell now becomes enucleated. This cell was fused with this cell. Now why they are fusing this cell with this cell? Because there are some proteins in this cell. Okay, in the normal process of sexual reproduction, how a new offspring is formed? When the sperm fertilizes the egg. Okay, in that process there are some proteins which are present in the egg cells which are required for the fertilized cell to multiply. So that is why, so there are some proteins in the egg cell which are required. So that is why they are fusing the egg cell with the udder cell, but they are removing the DNA from here because this is cloning. Only the DNA of one parent should be there. So then when they fuse these cells, and then they give the electric shock, and then this cell starts dividing. Okay, and when this cell starts dividing, from 1 it formed 2, from 2 to 4, and then they implanted this zygote into another sheep which acts as a surrogate mother, and then the sheep dolly was produced. So this is the way animal cloning is done basically. So the key point here is, in animal cloning only the DNA of one parent is involved. Okay, they're definitely fusing with the egg, but they're removing the DNA from here. Only the DNA from one parent is involved. That is why the offspring which is formed is the clone of the parent. If the mixing of DNA from two parents takes place, then it is not cloning, then it is the sexual reproduction. So this is the way they produce this dolly, the sheep. Human cloning. So using the same technology, human cloning is also possible, but it is banned because of the ethical issues. And the last thing I want to talk to you in this lecture is forensic biotechnology, how this DNA technology can be used for forensics. Now, to solve all the problems, like if there is a murder, who killed this person, or to solve the paternity problems, this DNA technology can be used. The very first application of DNA technology in these situations is the most important technique which is used is called polymerase chain reaction. It's called PCR. So to solve all these problems, like if there's a paternity problem, or if there is a murder somewhere, you don't know who is the murderer. These forensic people, what do? They go to the site, they will collect the samples from there, like you can collect some blood, you can collect hair samples or semen, but these samples are very, very little. Okay, so from these samples, if you have to extract DNA, that DNA will be very little, but you can use this technique called polymerase chain reaction. Now what does this polymerase chain reaction do? The main application of PCR technique is that if you have a very, very little amount of DNA, you can, by this technique, you can amplify the DNA. Okay, and then you can do the analysis on that DNA. Suppose if you go to the crime scene and you get a very little tiny drop of a blood. Now from that tiny drop of blood you are going to extract DNA, and that DNA is going to be very, very little. So you need more. So what you can do, you can use this technique called polymerase chain reaction, by which you can amplify the DNA. Okay. And it basically works on the same principle as we just talked about how does DNA make its copy. But the only difference is this technique is done in the lab. Okay, so how does this technique works? Like, this is your double-stranded DNA. So first of all, the two strands will separate, okay, and then before a new strand can be made, there's a requirement of a primer. And what is a primer? Primer is a short stretch of DNA, because so these primers are present in our cells also. Okay, so this is a technique which we do in the lab. So there's a requirement of a primer, only then DNA polymerase can make a daughter strand. So in the next step, so first of all these strands are separated, then primers are attached, and then the DNA polymerase is going to make a daughter strand, and the same thing happens here. Okay, so after one round of replication, you start with one, you have two strands. You start with the second round of replication, you have two strands here, you can end up with four strands. So this is PCR. This is called polymerase chain reaction. This is the way how DNA makes, how DNA can be amplified in the lab, and this is very useful technique for in forensics, because if at a crime scene there is very little sample, you can extract the DNA from that sample and you can amplify DNA like this, and then you can analyze the DNA. And once you have the DNA, you have large amount of DNA. How do these forensic people analyze the DNA? The analysis of DNA is on the basis of some specific regions in our DNA. They're called short tandem repeats. Okay, short tandem repeats, they are unique in every individual. Okay, so in our DNA we have these short tandem repeats. So these are some regions in the DNA. Basically in these regions we have some repetitive sequences, and which are unique for every individual, and they are called like DNA fingerprints. Like every individual has different fingerprints, so every individual in their DNA they have some repeats called short tandem repeats. So these are basically short DNA sequences which are unique in every individual. Okay, so these people, they will collect the DNA, they will amplify with PCR, and then they will analyze the DNA for these repeats called short tandem repeats. They will collect the DNA from different suspects, and they can then compare their the pattern of these STRs, and then they can find the criminals, or even the paternity problems can be also solved on the basis of this STR pattern. Okay, so this is the DNA technology. So we learned three basic things: how genes are cloned, how animals are cloned, and how this technology can be used in the forensics. So that finishes this lecture.